Welcome to the microbial world with another fascinating story. This time I'd like to talk about how we can watch vectorial proton translocation. We have learned that this is a quite important process. Actually, it's the most common event in living cells. And a cell like E. coli is shuttling across the membrane 1 to 8 million protons per second. This helps to generate ATP. This is vectorial proton translocation. This is the ATP synthase. And the question is, when this is so often, so rapidly occurring, why is it so difficult to observe? And indeed it is. And the reason is that the proton shuttling is in a steady state and extremely fast. So once a proton has been pumped out, it will be back within a millisecond or a few milliseconds. And we have to use a pH electrode. We can use a pH electrode, but only in the outer bulk medium. It's not possible to measure this inside a single cell. They are too small, and the electrode would destroy the membrane and the transport systems. And we have somehow to break the steady state. That means we stop the respiration, we stop the electron transport, and then we add a little bit of oxygen, for example. And then it will run for a very short time and will pump some protons, and we can see what happens. Actually, it will happen too fast, and we have to do another trick, and that will be we have to slow down the process, and we can do that by neutralizing or destroying the membrane potential. Okay, so for experiments you need some device, and I have used very long time during my scientific career a multi-electrode device for watching bacteria at work. And this has an oxygen electrode at the bottom. You can also use it as H2 electrode if you change the polarization here at the bottom. So we can see oxygen coming in and disappearing. We have the pH electrode, very important to see any pH changes. I was working with sulfate reducing bacteria, and we measured with a sulfide electrode, which is actually a silver wire covered with silver sulfide, to measure sulfide production or consumption. And we had a platinum electrode, which is a redox electrode, and you can see any changes of the redox conditions in your chamber. Then the whole thing is filled and the cells are washed that we are using. They have quite a high density, and we saturate normally the whole suspension with H2 or with N2, and we don't use buffer. We have only KCl, so this is allowing very sensitive pH measurements, changing uptake and production of protons. And we have everything at a constant temperature, and we can even switch on light or switch off light if you are doing photosynthesis experiment with this. Everything can be done with this multi-electrode device. But actually what we need mostly is the pH electrode, and today for my tutorial, once we have the O2 electrode and we have the sulfide production as well. Okay, and this is how a typical stop flow experiment looks like. So the cells are incubated under N2 anoxically, and nothing happens, and we measure the pH very sensitively. So you see a scale from 6.95 to 7.05, and after some time we add a little bit of oxygen, just KCl saturated with air or with O2. And then you see it's a very tiny addition, only 0.1% O2 saturation. And this has been respired away within a few seconds because we have quite a dense cell suspension. But this is coupled to respiration and electron transport. And electron transport means proton transport, and you see a peak of protons coming out, and this is flowing back. There's no more oxygen after a few seconds, and then we still see protons coming back, and these are flowing back via the ATP synthase. And it's not a constant rate. It's so that in the beginning it's very fast. The bigger the difference to the equilibrium in the beginning, the faster the backflow. This is named first order kinetics. And actually, one never sees all the translocated protons, because as soon as the translocation starts, the backflow will start. And when it's the highest, the difference is the highest, the backflow is the fastest, and that means it probably will be here. And that is difficult to calculate. You have to extrapolate it or to do some simulation, as I will explain in a minute. So these are my very first published results on proton translocation. I did it in Amsterdam in the group of Adrian Stouthammer together with Henk van Verseveld. And I had aerobic cells, CO oxidizing bacteria, carboxydovorans, and they were incubated without oxygen. Then I added a little pulse, 10 microliter of air saturated KCl, and then you get some proton extrusion, proton translocation, and it's flowing back. And for calibration, we added 10 microliters of 1 millimolar HCl. Then I can see this is 10 nanomoles of protons coming in, and this is more than 10 nanomoles of protons coming in. And we did an experiment under CO on the left and under H2 on the right side. And you can see with hydrogen, there's more proton translocation, and the backflow is faster as well. So it's steeper here because we have a bigger difference. The calibration curve is here again. Okay, but you also see this is 1 minute, and this is maybe 1 or 2 minutes until it's more or less back. And this is much slower than we had calculated before, where we said there's 1 million of protons coming in and out in a second. And this is because we have slowed down the backflow, and this was done by doing something on the membrane potential. So this is the reason why more than 99% of the electrogenic protons are coming back, and we have to neutralize or destroy it, and then we can see the translocated protons. Then the backflow is only due to the delta pH, and this will be much, much slower. And we have a chemical, KSCN, or potassium thiocyanate. Don't mix it up with cyanide. That dissolves in aqueous solution to K plus and SCN minus. And this SCN minus, the thiocyanate, is a membrane permeant anion, and it can diffuse over the membrane and can transport charges, the negative charges, across the membrane. And we incubate it for 20 minutes, half an hour, with a high concentration, 100 millimolar, for example. And then it's everywhere. And when there is now electrogenic process going on at the membrane, some of the SCN minus will cross the membrane and compensate for that. That means the membrane potential is not existing anymore, but the delta pH is still there. Okay, then there are other things we have to look at. This was not the case with hydrogen. This is again the same peak I've shown a minute before with hydrogen. And if you look at the reaction, it's 2H plus 2H2, sorry, plus H2, goes to water, and there is no proton involved. There is no proton produced or consumed. And that means you have a reaction, and after everything is done, you are back home to the same value as before. And this might be different if you have other metabolic reactions that consume protons or produce protons, and if you have transport processes that also might consume protons or produce protons. And this is especially the case with the sulfate-reducing bacteria. And we call these changes, if they are occurring, scalar, because they are permanent, and they change the amount of protons in the system, while the vectorial proton translocation only brings it from one side of the membrane to another side. Yeah, I have to come to the sulfate reducers, and this now becomes a little bit more complicated. I'm sorry for that. And when I went from Amsterdam to Konstanz to the group of Norbert Pfennig, I had a free decision what to do, and I decided, oh, okay, I just learned how to measure proton translocation. Let's try this with the sulfate reducing bacteria as well. And instead of adding O2, I just added sulfate. And then I get curves like that. And here the H2S formed a little bit later, so it started a little bit later. And instead of giving an acidification, what I saw was alkalinization, uptake of protons. Yeah, I was very fascinated by that, and I thought, might it be that I have discovered a new type of bioenergetics where everything is reversed, so they are not excreting proton, they are taking them up. And actually, after thinking about it a few hours, I found out, no, it's naive what I did in the beginning. I have to think about sulfate transport. So definitely sulfate reduction occurs inside of the cytoplasm, and the sulfate has to be taken up. And maybe what I see, and indeed what I see here, is sulfate transport. And then you don't end up with the same pH as before, because sulfate reduction causes scalar alkalinization. That means there are protons taken up. And sometimes you might see a little bit vectorial proton translocation here in trace B, but it's difficult to understand these curves. And that means I thought about making a simulation program named proton-exe to simulate these processes and to understand how everything is going on simultaneously. And look at this curve here, and this is what the proton-exe can make with it. It can even do more, but it's in the first very beginning. It's the same curve now, and we can understand the processes going on if we assume there's sulfate uptake very rapidly. It's added here to 10 nanomoles, and then it's consumed very, very fast. And with some delay, there's H2S production. We have measured it, as I have shown it, and we can simulate it as well. And while the H2S is formed and released from the cell, we get some alkalinization. And overall, we get an alkalinization due to the fact that sulfate reduction causes proton uptake. Yeah, and sorry for the busy figure here. These are more proton-related processes in the disulfide vibrio cell. And I would like to shortly go through the single steps. So we have the uptake of sulfate, and this is in symport with protons. It might be 2 protons. Then this whole thing is electroneutral. It doesn't change the membrane potential at all, because there's no charge coming in or going out. The cells, most sulfate reducers, when they are living under sulfate limitation, start to induce electrogenic transport systems. And that means they use a third proton, and that helps a lot to take up sulfate and makes it faster. And this makes use of the membrane potential. Then inside the cell we have the sulfate reduction. Sulfate does not have any protons, and the H2S formed carries 2 protons, and this might be the 2 protons that just have been taken up together with sulfate, and the release of H2S can be by simple diffusion. So you don't need a transport system like here, here, and here. This is a gas and neutral and not charged, and it can just leave the cell, and then outside it will dissociate to half H2S and half HS minus plus half proton at pH 7. This is the pK of sulfide to H2S. And that's when we see the release of H2S is going on. Then we have the ATP synthase, already known. We have an electron transport system that translocates protons. And we have even one more interesting system, and this is that the hydrogenases in many desulfovibrio are located in the periplasm, so outside the cytoplasmic membrane. And when the hydrogenase oxidizes H2, it forms protons, which are staying outside of the cytoplasm, and channels via cytochrome C the electrons into the cell. That means there's an electrical charge separation, not by proton translocation, but by electron translocation. And this is named the vectorial electron transport. Yeah, all of these things can be seen, and that makes it really interesting to make a simulation of that. And here is again what the simulation program ProtonExa can produce from that. So we have in this case the electrogenic transport, very rapid. We have the intracellular sulfate reduction consuming protons, and then we have the release of a few protons if the H2S that is coming from the cell is dissociating to H plus and HS minus, partially dissociating. And finally, we have the alkalinization due to sulfate reduction as expected. The simulation program, ProtonExa, will be explained in the next tutorial.